and it looks like we are recording. So, okay, so before we get started, Jen and I will be talking about case control studies today, kind of from the perspective of a curator rather than from a clinical epidemiologist or a statistical geneticist. However, we do include resources that are, for instance, clinical epidemiology textbooks to get an understanding of general concepts of what you may end up seeing in case control studies, and a lot of the different statistical significance values that we've actually seen through different papers that we've curated. So I've listed the references at the end of this talk, and I think we're going to actually put these slides up on Confluence after the call, so they'll be readily available. And additionally, if anybody has questions after this call for us in studies that they've looked at, we're happy to help as well. Or I can at least say I am. So what we'll be talking about today is this section of the genetic evidence matrix that focuses on case-controlled data. And so I'll first go through a little bit of an explanation and background behind single variant analysis and aggregate variant analysis and what those things mean in the context of the SOP and also the clinical validity framework, and then the criteria that we generally kind of look at when we assess case-control studies. And then the point values usually range on the order of 0 to 6 with a maximum of 12 points that can be given for case-control studies. And really the reason we use this is so that this allows for flexibility for the curators and experts when they're looking at data in the way that they want to score studies. So to start out, case-control studies are usually composed of 2 cohorts. These are essentially cohort studies. For instance, the case cohort are usually composed of individuals ascertained from an institute, an academic medical center, a diagnostic lab potentially, etc., or from multiple centers based on a given phenotype or disease. But the individuals in a case cohort could also be part of a defined population at risk. So these could potentially be healthy individuals, but the study could be looking at lifetime or cumulative risks or relative risks for particular variants in a gene or a single variant in a gene, such as CHEK2-1100LC. And these case cohorts are usually based on clinical indicators of hereditary predispositions to a particular disease, unless the individuals already are affected that they're being looked at. And so what I mean by in this point is that usually when individuals are ascertained within these studies, from a lot of the studies that we've seen, it's been based on them potentially having a high prior probability of potentially being predisposed to that disease or being affected. And this is usually based on their family history or they've been affected at a young age of onset. But there's also studies where the case cohort is completely unselected, meaning, for instance, all comers that have colorectal cancer, regardless of their family history, and only based on the fact that they're affected with that particular disease. For control cohorts, these are usually individuals that are typically unaffected, and they serve as a comparison group to the cases. They're non-cases in this case. Some studies will ascertain controls that lack the phenotype or disease of interest, but may not otherwise be unaffected for other phenotypes. And what I mean by this as an example is cancer-free controls that may have been drawn from a population database that was originally ascertained on cardiac phenotypes. Additionally, relative risk analyses may include relatives of probands that we've seen in some papers. And these controls may or may not be matched to the case cohort, and I'll go through in a little bit how controls can be matched to the cases. Additionally, meta-analyses exist, or single meta-analysis. You may find in your curations where the case and control cohorts are typically drawn from multiple studies and published as one large set. And there's usually specific statistics that are done to look at the pooled effects of all of those studies together in terms of how likely a gene is to put cases at risk or to be associated with the cases versus controls in terms of variants in that gene. So for the quality criteria that we address, and before I go any further, feel free to interrupt me at any point if something doesn't make sense or if you have questions. So for single variant analyses and aggregate analyses, by single variant analyses, we're essentially talking about when an individual variant is looked at in a gene and cases and controls, and then the statistical significance is addressed for that individual variant. So an example of this could be looking at an odds ratio for check to 1100 del-C in 20,000 breast cancer cases and 15,000 controls. An aggregate analysis is essentially taking all of the variants in mass, and this could either be 2 or more variants in cases and controls, and looking at the statistics in terms of how many cases and controls have either one of those variants or all of the variants. An example of that could be looking at the odds ratios of variants in, as an example that I'll talk about later, RNF43 in individuals that have polyps versus individuals that don't. So that can be an aggregate analysis where the statistics is looked at for all of the variants in that gene. So for variant detection methodology, this is one quality criteria we look at where we ask the question, were both cases and controls sequenced for the entire gene under analysis or genotyped for the variant under analysis? And what we mean by this is if you end up seeing a study, for instance, where all of the cases were sequenced for a particular gene, one loss of function variant was found, and then that particular loss of function variant was then genotyped in a bunch of controls, and they ran statistics on the controls with the one variant versus the cases out of the one variant, those would not be matched because there may be other variants that could exist in the controls that would increase the control count and potentially cause a statistically significant association to no longer be significant. And so those would not be matched. But if all the cases are sequenced and all the controls are sequenced, or all the cases are genotyped and all the controls are genotyped to the same variant, those are matched. And so that's why that matters, because it can end up changing essentially the numerators of fractions that are being compared later for statistics. For the power, here we're really drawing on whether or not there's been a sufficient number of cases and controls ascertained or assessed within the study. This really depends on the prevalence of the disease and the allele frequency of the particular allele that's being looked at, as well as the effect size of the gene being analyzed. We've been starting to see breast cancer cases and controls or case control studies where there's upwards of 60,000 breast cancer cases looked at, and compared to EXAC, for instance, as 65,000 controls. But we've also seen studies where there may only be 15 or 18 people looked at for, I don't know, polyps compared to someone with controls. And so it really depends on the prevalence of the disease in the population and the other factors that I discussed. For bias and confounding, what we assess here is whether controls are matched to cases based on ethnicity, location of recruitment, genetic ancestry, age, or gender. And in terms of genetic ancestry, the way we've seen this expressed within papers is if they've done an exome analysis of all the genes in someone's genome, they may take common SNPs within those people's genome and map them back to the hap map or the haplotype map and essentially determine the exact genetic ancestry of those individuals. And then they may do the same for their controls. If their controls have undergone exome sequencing and basically map them according to European genetic ancestry, for instance. But there's only been, I think, one study that I've seen so far that has done that. Cases and controls, whether or not they're equivalently evaluated for the presence or absence of phenotype and family history. And this can sometimes be an issue, for instance, when EXAC controls are used in terms of the presence or absence of a phenotype, even though we know the cohorts for which those individuals are drawn on, we don't always know if they were directly assessed in the same exact way as the cases in terms of their phenotype. And so that could be something to think about, as well as what the family history may be for those individuals that could already give them a higher probability of potentially having some sort of pathogenic allele. Additionally, was previous testing conducted on cases? We've seen case control studies in the past where maybe due to time where a gene hadn't been discovered and associated with the disease yet, or simply they just didn't test known genes from that disease that we would expect to be definitive. When looking at cases in that, and actually controls as well, controls may not have previous testing, to be honest, but when looking at cases, this can particularly matter just because there may be another gene variant that could explain that person's disease that we just don't know about. And then additionally, were cases ascertained due to being from families with the disease history, this can bring in a bias in the form of ascertainment bias where the particular individuals may already have a greater I don't know, genome makeup in such a way or genetic background per se that predisposes them to disease, and so therefore the allele being looked at could end up having a smaller effect as a result. Whereas looking at unselected cases are really looking at anyone with all sorts of genetic backgrounds for this particular variant. And so I think what we tend to see with this in selected cases is that can sometimes inflate the risk estimates. For statistical significance, we recommend weighing this value carefully, where individuals really consider the strength of the association. And so, for instance, if multiple pairwise comparisons are done looking at 5 separate genes in a case control study, we've often had to dig into the statistical methods of the paper in the methods section to really make sure that multiple test correction has been done. And this is usually in the form of a Bonferroni test correction, where essentially a p-value, for instance, of 0.05, if that's used for statistical significance cutoff, it's just divided by the number of cases that are looked at. And then that is used as the statistical significance cutoff for the paper. And then lastly, in terms of cohort composition, something to be careful of is if cases overlap across studies, be sure not to double count those studies when scoring. So for instance, we've seen from breast cancer consortiums meta-analyses that have looked at check to 1100 del-C. And that meta-analysis may have had encompassed 5 or 6 earlier studies, so we made sure not to count points for the 5 earlier studies, but just count points for the meta-analysis. For measures of association and risk, one of the things that we often see is the odds ratio, which essentially measures the frequency of variance among cases versus controls. And in this case, a stronger association between variants in a particular gene and disease results in a higher odds ratio. In terms of thresholds or cutoffs for when or when not a curator should count points for a study, the hereditary breast and ovarian cancer and colorectal cancer polyposis gene disease curation teams use a cutoff odds ratio of 2. And I think that has something to do with 2 is considered clinically significant, I think particularly in the breast cancer field, and provided the 95% confidence interval that's given by the authors does not cross below 1, and a p-value exists that is less than 0.05 over 0.001. And this really depends on the study, and also what these cutoffs will be disease specific. And so we recommend definitely talking to your experts in terms of, okay, what are we looking at? What's the prevalence of the disease? What would we really expect for an odds ratio cutoff in terms of what we want to assign points to or not assign points to? One other odds ratio measure that we've come across is the Mantel-Heitzel odds ratio, and this is often seen in meta-analyses, which is a pooled odds ratio across multiple studies, and sometimes, often in the methods, we'll end up seeing a sentence that relates to a fixed effect model being assumed. I don't know as much about what that really means, but I can always look into it more and give you all more information if you're interested. For the Fish's exact test, this is essentially looking at the proportion of cases with variants and whether that proportion is significantly different from the proportion of controls with variants. It's similar to a chi-squared test, but it actually gives an exact p-value all the way out to multiple, I guess, decimal places. So for instance, we've seen p-values at 10 to the negative 26 in papers, where the chi-squared test, what I've seen in the past, as the p-value gets progressively smaller and smaller, it can be returned as just less than 0.001, for instance. So it can end up being a more exact measure. Additionally, relative risk studies are looking at how likely individuals with variants are to become affected relative to individuals that lack variants. For example, a relative risk of 2 could mean that heterozygous variant carriers are 2 times more likely to develop breast cancer than individuals that lack the variants. Standardized risk ratios are ratios of the weighted average of category-specific risks. And so this is essentially, for instance, looking at a ratio of observed frequency of pathogenic variants in cancer cases divided by the summed frequency of all pathogenic variants in the control population. We've seen this in a recent study we looked at for ovarian cancer. Standardized incidence ratios are the ratio of observed number of disease cases divided by the expected number of disease cases. And in this case, the expected number of disease cases is usually what might be expected if the population had the same experience as some larger comparison population. And I've often seen this described as NSIR of 2 could mean that there's a twofold greater risk in individuals with this variant to have disease versus the general population, for instance, because they think that the expected number is potentially representative of what we would find in the general population. The hazard ratio measures how often disease happens in one group, for example, individuals with pathogenic variants versus another group, for instance, individuals without variants over time. And so one factor that I'd be careful to consider here is how often follow-up occurs. One thing that we've realized in looking at hazard ratios for colorectal cancer, for instance, is that cases may have been followed up over time more frequently than controls. Controls may have been individuals that were, okay, you come get a colonoscopy every 10 years, and so there may have been individuals where they essentially, I guess, did not get the disease even though they were variant carriers simply because their follow-up time was longer than another because they forgot to go to their appointment. So the point that I'm making here is in terms of the time to follow up, that could potentially matter in terms of looking at hazard ratios. So now we'll go through a couple of examples of what we've seen in the literature to try to give everyone an idea of kind of how to deal with these. And Jen and I will essentially go back and forth between examples we've seen. But before I go any further, does anyone have any questions on what I've talked about up to this point? Okay. All right. So this is an example on breast cancer by Couch et al. That was published in 2017. And for this use case, I'll talk about ATM and the curation that we did for breast cancer. So this study was an aggregate variant analysis looking at 29,229 Caucasian breast cancer cases referred for essentially next-generation sequencing panel testing to Ambry. The cases were sequenced by NGS for 16 susceptibility genes, and they were compared to roughly 26,644 controls from EXAC, and these were specifically controls that were non-Finnish European descent, and they excluded any cases that were brought into EXAC from the Cancer Genome Atlas to make sure that it wouldn't have been biased in that regard. For variants, in terms of cases that were counted or controls that were counted, they were looking at those that were pathogenic or likely pathogenic by Ambry's classification system, and that classification system I actually think is listed on ClinVar for their submissions, and control variants in individuals in EXAC that were deemed pathogenic or likely pathogenic by Ambry's system, or control pathogenic and likely pathogenic variants that were nonsense frame-shift or consensus splice site variants that may or may not have been classified by Ambry or listed in ClinVar. Additionally, controls were included for individuals that had missense variants in ATM that were deemed pathogenic or likely pathogenic in ClinVar by clinical labs. So I think in this case, if it was OMIN that had submitted it and called it pathogenic, they didn't necessarily use those. So the factors that we considered in looking at this study before we scored it was that ATM was sequenced in cases and controls, so they were matched for variant detection methodology. In terms of the power, the total number of cases and controls was sufficient for breast cancer, and I'll go into a little bit more about this in terms of the control set specifically a little bit later. But in terms of the amount of cases they looked at, honestly, I think this may in fact be one of the largest breast cancer case control studies we've seen at this point. The total cohort itself was about 121,000 individuals, but the reason there's only around 29,000 cases looked at is those are the individuals that were sequenced for ATM or had been referred for testing by a panel that had ATM on it. The statistical significance, the odds ratio was 2.78, which is also listed here, and it had a 95% confidence interval, 2.22 to 3.62. So it met our criteria in that it had an odds ratio greater than 2, and the confidence interval didn't drop below 1. And the p-value for looking at the enrichment of variance in cases versus controls that I think was actually a Fish's exact test came out at 2.42 times 10 to the negative 19. So it was highly significant, and I think it was above their threshold based on multiple test correction. For bias and confounding, the controls were matched to the cases based on ethnicity because they looked at Caucasian breast cancer cases and European non-Finnish exact controls, but not necessarily genetic ancestry, location of recruitment, or age. But I think that may be a little bit harder to do because even though they could have looked at exact cohorts that their controls came from in exact, I don't know if they initially had ages on all of those people. But either way, we ended up giving this 4 points, and the reason we did that is in the SOP, I think it lists that if controls from population databases such as EXAC or the ESP are used as control sets, that we had listed something like 2 points for that kind of a study. The reason we decided to give this more is we discussed this as a group, and we felt that trying to find matched controls for 65,000 breast cancer cases out of the 121,000 individuals that they included that they listed in the study, but 65,000 of them were breast cancer in total. We thought that trying to find 65,000 matched controls could be potentially difficult, and so EXAC in this case can maybe make sense to use, and so that's why we gave it more points. And I think Jen has a colorectal cancer example now.
All right, thanks, Bryce. Right, so you can go to the next slide. So I'll talk about a case control study that we recently came across in our colorectal cancer group. This was a paper that was looking at over 1,000 unrelated patients that had colorectal cancer that had been diagnosed at 55 years or younger and who had at least one first-degree relative with colorectal cancer. So they had a family history of the disease. And these were UK residents with self-reported European ancestry. The control group was made up of over 1,600 healthy individuals from the UK. These were, I believe, 1 or 2 UK cohorts. And the one thing we thought was good about this study that Bryce had sort of mentioned in the beginning, and this was the only one that I've come across that did this, but they ended up using HapMap data to make sure that their cases and controls were matched by ancestry. And after doing that, they were able to exclude some individuals who had a non-Northern European ancestry. So these were really well matched based on genetic ancestry, location, and ethnicity. So you can go to the next slide. So they really did 3 types of analyses, and I'll only talk about one that we ended up using for our group. First of all, whole exome sequencing was performed in both cases and controls. So already we see that the variant detection methodology is well matched between the cases and controls. And the first analysis they did was actually a single variant analysis looking at moderately low frequency coding variants for an increased risk, an increased association with colorectal cancer risk. And they also added in some more cases and controls that had been genotyped. Bryce, I think there's something on the screen. There we go. Thanks. So they had added in about 5,500 cases and 6,700 controls that had also been genotyped for their single variant analysis. And they actually did not find anything significant, so that's why I won't mention the results for that one. But they next did more of a aggregate analysis, or in this case, a gene burden test, and they basically looked at the impact of rare alleles collectively within a gene on colorectal cancer risk by grouping the SNVs and indels for each gene and then comparing counts between cases and controls. So that's what we ended up scoring this study for. And then I won't mention, but they did do a third test looking for novel colorectal cancer susceptibility genes confined to 863 cases without a probable disease-causing mutation in a well-known colorectal cancer gene. So you can go to the next slide, and I'll talk a little bit more about the results from the second analysis. So basically what they did here was a T1 test, which is often used for looking at rare variants, and they looked at 3 different types of variants, so disruptive, predicted damaging, and then all coding non-synonymous variants together. And they set their significance at 8.0 times 10 to the negative 7th. So this was after a Bonferroni correction for the testing of about 20,000 genes. So they had a pretty low threshold for their significance. And in the figure down there, you can see we were actually curating this for MLH1, and you can see that MLH1 p-value fell just below their cutoff. So this was the gene that we ended up curating for. You can go to the next slide. So here are the things that we considered for this paper. So I already mentioned this, whole exome sequencing was done for both cases and controls. So pretty strong variant detection methodology. For the power, colorectal cancer is pretty common, so maybe having a larger study would have been more beneficial, but the experts did agree that this was a sufficiently powered study. As for bias and confounding factors, I mentioned that they matched really well by location and ancestry. Any controls that had developed cancer during the study were excluded. They also made sure there were no related individuals in the group. And they did match by age pretty well also. So controls would have been around 58 years old, and the mean age for the cases was around 47 plus or minus 6 years. And then for the statistical significance, they did do a correction for multiple comparison testing, and the p-value for MLH1 fell below that threshold. So we felt like this was a pretty good case control study, and we ended up giving it 5 points out of 6.
Okay. Thanks, Jen. Now I'll talk about an example that came up more recently with us for serrated polyposis. So in these individuals, this paper was from McGlaughon et al. In 2014, and we were looking at RNF43 for polyposis with this curation. Specifically, they were looking at 20 unrelated cases that were ascertained from Massachusetts General Hospital that had sessile serrated adenomas, which can essentially predispose individuals to colorectal cancer that fulfill the modified World Health Organization diagnostic criteria for serrated polyposis. And down here is the criteria. It's essentially the individuals had to have at least 5 sessile serrated adenomas that were proximal to the sigmoid colon, or any number of sessile serrated adenomas proximal to the sigmoid colon in an individual with first-degree relative with sessile polyposis or colon cancer, or greater than 20 SSAs that were distributed throughout the colon. Additionally, for controls, they essentially drew on the 4,300 European Americans from NHBLI exome sequencing project as controls, and essentially just looked at variances in their controls in their cases that would have been truncating or potentially pathogenic within particular genes that showed up of interest. And so within, and particularly at this, one thing I want to mention is the sequencing was done by whole exome sequencing analysis in both the cases and the controls. And in the cases, there were about 555 genes that came up they thought could be potential novel candidate genes for serrated polyposis. And RNF43 happened to be the gene that had truncating variants in it, and biologically it seemed like it could potentially fit because it is a negative regulator of the Wnt pathway, which is also what APC falls within, that predisposes individuals to familial adenomatous polyposis when it has pathogenic variants in it. So they found 2 over 20 cases with RNF43 truncating variants, 1 of the 4,300 controls with RNF43 truncating variants, and they used a cohort allelic sums test for looking at statistical significance. That was employed to assess the enrichment of strong loss of function mutations of the gene set in cases compared to controls. I drew this directly from the text. I can look more into the cohort allelic sums test. I've seen this one other time, but it apparently is kind of some sort of pooled analysis. So even though this ended up being very, very few cases, one thing that we kind of ended up looking at with this was really the power. So RNA-43 was sequenced in both cases and controls, so they matched in the variant detection methodology. And the case cohort was really small, in this case only 20. However, when we had talked to the experts on the call, we realized that early on in the paper they noted that around only 20 to 35% of all colon cancers are estimated to be due to serrated lesions, whereas I think the other ones that are due to polyposis tend to be mostly from adenomatous polyps rather than serrated polyps. And then additionally, they note that about 1 in 3,000 individuals in populations primarily composed of European ancestry is thought to be the prevalence of serrated polyposis syndromes. So it's not necessarily that uncommon, but we were really wondering, could they have had fewer cases simply because the phenotype could maybe be not as common in terms of a genetic association. So the 555 gene comparisons meant that from their cohort allelic sums test, they needed a p-value of less than 9 times 10 to the negative fifth to be significant. And what they found was from these 2 of 20 individuals versus all of the controls that they looked at, they had an odds ratio of 460. And I'm going to try to look up relatively quickly to see how many of their controls ended up actually having variants. And I think they potentially either didn't find any controls or only one control that was actually positive for a truncating variant RNA-43. It was really, really very, very few individuals. And so we thought that maybe this odds ratio could be pretty high in this case simply because they had a ton of controls they looked at, which actually can happen to odds ratios. If more and more controls are brought into the study, this can basically significantly increase the chances of getting a high odds ratio. And so even though we saw this really high odds ratio, which could have related to having more controls, the fact that the confidence interval was so wide here on the order of 23 all the way up to 16,000, we figured that really getting this odds ratio may have just happened by chance with such a large confidence interval. And so the large control set may have inflated the odds ratio, and the controls were only somewhat ethnicity matched, but it was really just drawn on European Americans, and there wasn't much other discussion to it. And having this large confidence interval, what we ultimately ended up doing with this study is not counting it as a case control at all, and we essentially took the 2 cases and counted them as case level data. And so this is kind of an example of if you come across a study where the odds ratio seems to be way too high given the phenotype that you're looking at, or the confidence interval seems way too large even if it is above 1, that's definitely a scenario to flag for experts and discuss with them in addition to other studies you discuss with them, simply because the math just may look wrong. So Jen, I think, has an example for HCM now.
Yeah, so we thought we would include a non-cancer case control study. So we picked one from hypertrophic cardiomyopathy that we've used in the past. And you can go to the next slide. So this is really a study looking at cardiomyopathy in general, but we used it specifically for hypertrophic cardiomyopathy. And the cases come from Oxford Medical Genetics Laboratory and from LMM, and totaled, I believe, over 7,000, almost 8,000 cases versus the controls that were downloaded from the EXACT database. So this is a case where EXACT was used as the control cohort. And when looking for rare variants, they applied a minor allele frequency threshold of 1 times 10 to the negative fourth. And the reason they came up with this number is that when you look at the most common pathogenic variant in both of the clinical cohorts from Oxford and from LMM, this was a mutation in MYBPC3 that was found in controls in EXACT at 2.5 times 10 to the negative fifth. So if you find, so they set their cutoff at 1 times 10 to the negative fourth because variants more frequent than this in the general population would likely not be a highly penetrant pathogenic mutation. So you can go to the next slide. So for their methods, they were really comparing the frequency of rare variants in exact versus the cardiomyopathy cohorts. And for each gene, they looked at the case excess, which is defined by subtracting the proportion of individuals in exact with a variant from the proportion in the clinical cohorts. And then they also looked at odds ratio, which Bryce has already talked about. And then another thing they calculated was the etiological fraction, which is sometimes used in epidemiology and estimates the proportion of cases in which a rare variant in the gene was causal. Go to the next slide. So the table we're looking at here is a list of hypertrophic cardiomyopathy genes. And you can see, so they're split into non-truncating and truncating variants. So anything highlighted in black was significant. And also on the right is the odds ratio with for the hypertrophic cardiomyopathy genes. And it's hard to see, but genes with asterisk next to them were statistically significant by, they did Fisher's exact test, and they also did a Bonferroni correction. So I believe their cutoff for HCM was 0.0025. So anything with an asterisk was considered statistically significant. You can go to the next slide. So some of the things we looked at for this paper, for the variant detection methodology, they had used sequencing data from diagnostic labs for the cases and exome sequencing for controls. So, so robust variant detection methodology in both cases and controls. For the power, HCM is relatively common and is estimated at about 1 in 500 in the general population. But they looked at almost 8,000 cardiomyopathy samples, so this was a pretty well-powered study. The main thing we looked at were the bias and confounding factors, and Bryce has touched on this a little bit. When you're using exact as a control cohort, you have to be careful of this because they can't be closely matched to the cases. So for instance, we're unsure of the quality of phenotype evaluation of the controls, so determining whether or not they actually have cardiomyopathy. And then for statistical significance, as I mentioned, they did Fisher's exact, and they did do a Bonferroni correction. So in this case, the statistical significance was sufficient. So we ended up giving this study 2 points for most of the genes we used it for, and as I mentioned, that's mostly just because of the fact that exact was used instead of a matched control population.
Thanks, Jen. So that was kind of most of the examples that we ended up having. I can always send around a paper or add an addendum. I'll probably add an addendum slide to this that just shows a table of what meta-analyses may end up looking like in terms of mental hypo odds ratios, but it's essentially very, very similar to many of the case control studies we discussed here that show odds ratios. It's just usually odds ratios looking at multiple case control sets, and as long as they don't necessarily overlap with each other, it's okay to count those in lieu of earlier studies or potentially other studies. In regard to that, you also may want to pay attention to the method sections because they may actually describe where they excluded cases from some of their internal case control sets they're looking at because they were covered in another study. So with that, these are the references that we kind of drew from, and they'll stay as part of the slide if you want to look more into this on your own. But are there any questions at this point?
Guys, that was great, especially for our hearing loss. We don't have a lot of case control studies, so this helped me a lot going through these examples. I'm just curious if you guys can comment on when case control studies have been enough to dispute a gene disease association.
We actually had that discussion yesterday at one of our UNC meetings, and we thought in the past, like this came out for BRIP1 and breast cancer, I think, actually. I don't know if Chrissy Lee's on the call or not, but she may know this. We've seen like, Christy, didn't we see 2 studies for BRIP1 where they consistently were not seeing enrichment?
I think there were 3.
Okay. So when there's been a lot, we've definitely considered it, but something that I kind of remember that I've heard before is that evidence of absence doesn't necessarily always mean, absence of evidence does not always mean absence of evidence does not always mean evidence of absence of an association. And so by that, even though there may not be an enrichment in a particular gene for a lot of pathogenic variants versus controls simply because that gene just has really, really low penetrance, as an example, there could be families that still exist where variants in that gene clearly segregate within those families. And so I think the 2 of those things kind of have to be weighed together in terms of making that call for disputed or refuted. I think in the case of BRIP1, we didn't see a whole lot of case studies that were showing, okay, clear segregation within families. Yeah.
Right. So it's a combination of nobody was really finding an association, and then several studies just were really well done and completely did not see an association. This was fantastic. I'm so glad you guys put this together.
Thanks. Glad you liked it.
So this is Jessie. I had a question for you guys about phenotype information. So some of the conditions that we're curating on a control population, might not be well phenotyped. We might have some, polyps might be under-recognized, and people with polyps might sneak into a control data set, or, the age of onset for the cancers involved might be, higher than what's, known to be for the control set. Like we know that there are plenty of folks in XAC or NOMAD that are in their 20s or 30s. Do you have any thoughts about maybe how to adjust or maybe give less points when the phenotype isn't one that could be, or one that's not for sure early onset or might not be picked up in the controls?
Yeah, I think with HCM, that's a good point because it can have a later onset. So that's why we were sort of towards the lower end for that study, only giving it 2 because given that the age range and XAC could be much larger, and you can't really know that these people won't have HCM. So that's why we definitely reduce points in that case.
I echo Jen on that, is that if there could be an age-dependent penetrance issue, especially in the controls, then, yeah, taking that into consideration and giving less points could really matter, because you're right. If they end up becoming cases later, it could either increase the statistical significance, or if they don't, maybe keep it the same or not. So, yeah, not having that information, and particularly with phenotypic assessment, can really matter as well. So yeah, I would say that potentially weighing that in terms of how many points you give can definitely be a consideration.
Yeah, we set our odds ratios for cancer maybe a little higher than some people would to try to get around how common like breast cancer is, for instance. And Jessie, I will also say sometimes it works against your case that the controls you have basically no information on, because some of the controls with your variant may actually have cancer and you don't ever know it. So I think it works both ways. I'm not sure it evens out, but not having that information certainly plays a role. And we've never given a study a 6.
Yeah, I don't think so. I just can't remember if we did. So there was this POLB2 Anthony paper that did I remember Fergus brought it up, actually, Christy. They showed the Bode-ICEA model for their relative risk studies. He said he was pretty impressed by it. I think he had it secluded, but yeah, I can't remember it. That's the only one that I can think of that we've maybe given 6 points to because of that model, but I'm not 100% sure on that. I have to check.
So any other questions for Bryce and Jen? So I'm just curious, for those of you on the call, I mean, how many folks are routinely or are looking at case control studies or have seen case control studies? I mean, I know the folks in the cancer group, and I guess for HCM we have a couple of case control studies, but has anyone else had experience looking at these for gene curation or variant curation? And any other thoughts or comments?
Hi, Jenny. I had a question. This is Courtney. Considering the fact that sometimes, you know, you will have a case control study, but it is not necessarily negating that the individual that they are including or individuals they're including are negative for mutations in other known genes that could be pathogenic for that disease entity. How would we just weigh that, again, in a conservative approach if we're going and they're actually showing several patients to have mutations in genes that are known to also cause the same disease entity?
Yeah, Bryce or Jen, do you have any thoughts on that? I mean, I guess what you're looking at is just the frequency of variants in the gene of interest in your patients versus controls. I mean, I see what you're saying. Like for HCM, you know, have they ruled out all the sarcomere genes, for example, if they're looking at something more rare, but you're still trying to see whether your particular gene or variant of interest is just increased in frequency in the cases versus controls. So, I don't know, Jen and Bryce, I mean, do you take that into consideration, like for breast cancer, and Christian, you're out there too, would you want your cases and controls to have been screened for like BRCA1 and 2 before you start looking at another gene? I mean, would that cause you to change the scoring, increase scoring?
That's actually mattered for us in the past with breast cancer, I could say. I think we actually found a case control once where they were looking at, I don't know if it was MSH6 or MLH1 or something, but one of their cases actually had a BRCA1 pathogenic variant that would have had to have been struck from the case cohort. And so we ended up, I think, not using it in that instance just because as curators we don't necessarily calculate odds ratios ourselves. So sorry to interrupt, Jen.
So are you also talking about like previous testing in the controls too, or just cases? I'm not sure if I caught that.
I was talking more so to the cases, but I guess you bring up a good point that you could equally have individuals in the control population that have pathogenic mutations as well.
Right. Okay, yeah, I wasn't sure if I heard that right, but yeah, because I was going to say I'm not sure how common it is to have any previous testing in the controls, but okay, yeah, I see what you mean now. Yeah, I definitely think the previous testing in the, if you're looking at, like, for instance, if we were looking at, you know, MLH1, but we didn't look at the other Lynch syndrome genes, I think you would have to weigh that because, you know, you don't know if they have mutations in the other Lynch syndrome genes.
Yeah, I think we're benefiting now. The bigger studies are mostly done at the larger labs on these cancer panels, and so they're getting all the genes of interest that we'd want to have looked at. So we probably don't have as much experience with having to dock some point. I can't think of any of our studies where the folks that were looking at breast cancer risk where BRCA1 and BRCA2 weren't looked at. In fact, some of them now exclude those carriers and look at folks that have an unexplained cause after BRCA1 and BRCA2.
Yeah. We've seen that same thing with the MMR genes in APC and UAH where they're excluded.
Yeah, that said, I know with the HCM it's been hard because generally they're just looking at individuals diagnosed with HCM, but they're not excluding that they have the mutations in the 8 well-known sarcomeric genes. And so oftentimes if I look in the supplemental evidence for a more rare gene that's not one of the 8 sarcomere, I can find that they have an MYBPC3 or MYH7 pathogenic mutation or likely pathogenic. And so normally as a case study, like or individual report, I wouldn't count it, but now all of a sudden it's counted in this odds ratio. So it made me wonder how we would score if we see that there's no indication that all these individuals were negative for no mutations.
Yeah, it's a good question. I guess I'm thinking if a variant isn't pathogenic anyway, you wouldn't expect to see an enrichment in the cases. So just kind of playing the devil's advocate and looking at it the other way around. I mean, if you truly are seeing enrichment of a variant in the cases, I mean, either it's having an effect or it's in linkage disequilibrium with something that's having an effect.
And sometimes we'll see that if there's like SNP association studies that we come across.
Right. So are there any other questions? This has been really great. Thank you so much, Bryce and Jen. We have just a few minutes left, so any other questions, by the way, or, you know, I'm sure Bryce and Jen will take questions afterwards as well.
One other comment I would make is when we're talking about these, we're really looking at these from the perspective of diseases where we're thinking there's a Mendelian inheritance component to this, not necessarily association studies looking at more common SNPs. Even though we've come across as in curations, we haven't usually scored those or included them in our curations. Unless I guess it's a really, really well-known SNP and it's known to be high in the population, but it's also known that that SNP definitely causes disease. So.
And maybe, and Bryce, I know you brought this up at the beginning, but also always with these studies, just being careful not to double count. So we've definitely seen with HCM, you know, the same case cohort being used in multiple studies. So just making sure that that data is only being used once in your curation. So you're not counting that study multiple times. So just being careful about that as well.